Hi, I'm just going to give you an update about the RHDV release. So RHDV is a virus that affects rabbits, and RHDV1, which is the one I'm talking about today, affects only rabbits. And we've been using a biocontrol agent in New Zealand since it was first released in about 1997. But since that time, RHDV still does control rabbits, but in some areas, rabbit populations were starting to increase again, and so we were looking for better versions of RHDV to release, and at the same time the Australians had identified a new version of RHDV called RHDV1K5, and that's basically what I'm going to talk about today. So RHDV1K5 was a potentially significant tool for rabbits. It's not a new virus. It's a Korean strain of the existing RHDV1. The original strain was called a Czech strain because that's where it was originally found, and it is still present in New Zealand and was widespread before the new K5 release. Now, K5 was selected in Australia for a couple of reasons. It's the most important ones that it can overcome immunity provided to rabbits by a benign strain of rabbit calicivirus. So rabbit caliciviruses, there is about 4 different classes, and there's these benign ones that are found in every country, and they act like a vaccine and protect against the pathogenic strains. So K5 could overcome immunity due to that benign strain. It was also quite a pathogenic strain in its own right. It didn't take many virus particles to cause an infection, and it had potential for improving the biocontrol system. The Australians and ourselves, we made clear right from the beginning that this was not going to be a silver bullet for eradication. Biocontrol seldom is, and in fact basically never is, but as part of a long-term integrated approach, it was a useful tool. We weren't expecting to see the big knockdown that we saw when the Czech strain arrived in 1997 because the rabbits are no longer naive. They have seen these caliciviruses, and they have some immunity, and also the new virus was going to have to compete with the existing strain. What we expected to see was an improved knockdown by about 25 to 40% on average, but we knew there would be a large range between 0% in some areas and 70% in others, because that was going to depend on the population and the climate, and also on the number of susceptible animals in that population. So the K5 release, and I'm cutting out a lot of stuff here about regulation. It took us nearly 2 years to get this approval through. And we piggybacked on the Australian experience. They did a lot of the non-target testing and all the things that we had to do, and we were very grateful for our Australian collaborators, which allowed us to provide the information that EPA and then ACVM required before you could do such a release. The plan was to select sites, and these were selected by our local councils, and we were recommending that they had at least 100 rabbits within 100 to 150 hectares, so they had to have a decent rabbit population, and we recommended that they could release it at spacings of about 20 kilometres, because we thought that over a 3-month period the virus, which spreads quite rapidly for a virus, would spread 20 kilometres over a 3-month period. We recommend an autumn release because that's when the number of young rabbits around is low, and also there are plenty of fly vectors present to move it around. The thing about the RHDV1 class of virus is young rabbits have a period of protection up to 8 to 10 weeks where they're not susceptible to the virus. And that's probably how the virus survives, because if an animal does get infected, an older animal does get infected, and they have got no immunity, then that's a naive rabbit almost always dies. So we're talking 95% lethality. The other thing we were stressed in our release plans that we prepared, and operating plans with the various regional councils, that pre-feeding was a very important part of this one. We used untreated carrot. Generally carrot. There was some people used oat baits, but that was quite rare in the New Zealand situation, and this was essential to ensure that rabbits were used to eating carrot, and would come and take the treated carrot and have a high uptake. And this became particularly important in some areas of Otago and Canterbury, where they had unseasonal rain in the February area of over 100 millimetres in several areas, which meant instead of going into an arid region looking desert-like, we were actually walking through 4 to 10 centimetres of lush growth, which is most unusual in the places which normally have rabbit populations. We were using a high quality commercially prepared product, and we were trying to avoid an illegal introduction of the RHDV1 K5 because there were biosecurity issues. Australia has other viruses and other diseases that we did not want to be mistakenly introduced if someone did an illegal introduction. I'm sorry about this graph, it seems to have resized itself. With these new releases, we were looking at using it as using natural spread. So that in the past, people had used it as a biocide, where they'd prepared tons of bait and they had spread tons of bait over the landscape. So animals would eat the bait and they would die rather quickly. And that's what you see when every individual gets exposure at one time. It's too expensive to do that, and we felt that natural spread would give us a better chance of getting good establishment. And so we were expecting to see something more in the shape of this blue sign. So we expected to see the individual animals that got exposure to die and then provide a source of inoculum that would spread to other animals initially in the local area and then wider afield. So we expected to see a lower peak, but a longer period where RHDV K5 would be spreading in the environment. And as I said, we were basing our data on expecting a spread of about 7 kilometres per month, and that would go for maybe 3 months while the fly populations were present. And this was based on basically on half the rate of spread that was seen in Spain and half the rate of spread that was anticipated for Australia, because New Zealand has a different climate and we thought we might have different vector populations. So we set it as a conservative number. So the release and distribution day came, and the first release was actually we got approval in February 22nd, I think, of February, and in 18, and we got it imported the first week of March, and the first release was done on the 12th of March. There were 450 vials imported, and they were taken up by 8 of the 12 regional councils and 2 of the 4 unitary authorities. And these were basically the areas which had rabbit problems. Some places did not participate because the impact on the rabbits was considered to be negligible, because rabbits don't do very well where it's very wet. And in total, in these first 6-8 months, there's been 271 releases, and some of them are still ongoing, like Auckland Council will release theirs next year on some of them, their maunga. The thing about this release is you can do control where you can't do shooting or toxic control. It's got a much safer human safety profile. And you can see the breakdown in the regions. Most of the places where it's been released have been Otago, Canterbury, and in places with arid areas. And this is the map for Canterbury. You can see it's concentrated. There's a little bit up in the north, Canterbury, a bit peri-urban areas around Christchurch, but most of it is in the Mackenzie country and in that part of Canterbury. So I'm just going to talk a little bit about the results from these field releases, and then I'm going to talk a little bit about what we found from the science. So this is the results of Canterbury night counts over 6 to 8 weeks after the release. They did spotlight counts, counting the number of rabbits over 129 kilometres and 4 high country stations, and the average reduction in rabbit numbers for over that 6 to 8 week period was 40%. Now we don't know what every individual rabbit died of, but it reflects the impact of K5, the Czech strain, the original Czech strain, and also there'll be some natural mortality and traditional control like shooting. But it is a significant reduction, and it was pretty much within our ballpark, as good as we could have expected. And on the property basis, there was about a 0 to 70% reduction. So in some places it had no impact, and that's probably because the Czech strain was particularly active this year. There was a lot more vectors because there was a lot more moisture, and in a lot of places we went to in these areas, the Czech strain had just gone through the rabbit population and had left a very high level of immunity. But we still did get some, we were very pleased with the response, and this data is from Grant Sullivan and Brent Glentworth from ECAN. So alongside of this, we were looking at some research, and we wanted to understand how RHDV moved. Rabbits actually don't spread very far in their day-to-day movements. They don't go long distances, and the rate of spread of RHDV has always been a lot faster than the rabbit movements. And so it's been known for some time that flies play a very important part of this. So we set up 4 science study sites, 2 in Otago, 2 in Canterbury, and we had a central release site, and then around each site we had 10 satellite sites, and they were between 1 and 5 kilometres away from where we did the actual release. And we just monitored weather, we looked at fly abundance, we collected carcasses and looked at camera trap setups, we looked for the presence of virus in the flies and the carcasses, and we recorded the impact on rabbit numbers with spotlight counts. And today I'm just presenting you preliminary results. So these are our 4 science sites. There are 2 in Canterbury, 1 at Tekapo, 1 near Pukaki. There's 1 up in the Ida Valley and another 1 in the Cardrona Valley. And these are all fairly rabbity properties. So at each science site we had this little release site, and then we had these 10 satellite sites where we did all our measurements. And that was that's a 5 kilometre circle, which meant that we had to set up our sites at least 10 kilometres away from any other release site, and that limited to where we could put our sites, because if we had our release site closer than 10 kilometres, we were likely to get virus moving into our release site from other release sites. So in that central site, traditionally when you release RHDV on a bait, you'd just do a strip going 2 kilometres, and that would take 10 kilos of bait and you'd spread it over a 2 kilometre. Right, but we wanted a spot release, so we just did we just walked a line about just under a kilometre, and we just released it in this concentrated area so that we could see how the virus moved. And then what we did was just monitored these yellow fly traps, and we tried to monitor the movement of the virus to the fly traps, and we were also monitoring the movement of the virus out to these satellite sites. So I showed you that nice layout thing, but in reality life is not like that. And so this is an example of an actual release site. This is Glentana. There's our release sites, and these yellow triangles are our satellite sites. And of course they can't be in a nice circle because we've got a lake here, we've got some rabbits over here, but we couldn't really get to them because there's a strong river, and this area of land doesn't have a lot of rabbits because of altitude. So in actual fact our satellite sites had to adapt to the environment, and we can see the pink dots were where we found rabbit carcasses in the following 8 weeks after the release. That's our miniature weather. There's our fly trap, and that's what we were hoping to see a lot more of, but finding dead carcasses proved to be quite tricky. People were out there for 20 minutes every 3 days searching in the satellite sites and in the ground zero site, and we did find some carcasses, but we had hoped to find more. What we did find, however, was we had all these fly traps, and we were able to look for RHDV and flies. So this is a week before K5 release, and these are collections from fly traps around the release site, and this little bit in the here are just some standards. And you see these bands lining up to this standard, that indicates it was RHDV1 check. So there was RHDV1 check in all the sites and just about 80% of all the fly traps before we did the release. 1 week after release, again we still got quite a lot of check, but we're starting to see, well 3 days actually, we're starting to see K5 appear, and we did find K5 in all our release sites. What I've got now is I've got over 2,000 samples from fly traps to still analyse, and it's not a fast process, and we'll be able to follow not only how the fly trap's further away, whether they've got the virus, and we'll also be able to see whether the virus persists, whether it stays there for the whole period. So we were able to detect it in flights all prior to the release, and post-release we also got the Czech strain, and I'm still looking at those samples because we're going to use that to look at rate of spread and persistence. So it's a very simple system. It's just a fly trap with some rotten carrion meat that attracts carrion flies, and then we sample from the fly trap. So as I said, we did find some dead rabbits. Clintana we found 27, but actually a lot of them were either old or had been shot, or we just couldn't detect virus. So the results were, what we found is that of the 43 recovered carcasses over the 4 sites, excluding the very, very old and the very new, we found carcasses which had Czech prior to release. We found K5 in 3 out of the 4 sites after release, and we also found some Czech in half the sites still after release. So the one place we didn't find carcass was in the Ida Valley, but there was plenty there in the flies, and what we did find in our camera studies, there was an awful lot of predators present. So there's 2 things going on. We're not seeing the huge die-off that we saw in 97 because it's not a naive population. What we were seeing is an awful lot of, and when the carcasses were there, the predators were pouncing on them and removing them. We could see where they'd been some places, but we couldn't get good samples. What we could see from our fly traps and from actually this is just from the carcasses, we could see the virus was spreading about 53 metres to 375 metres per day in our science sites. And that's just a preliminary result, so that's looking at about 1.6 to 4 kilometres a month. So as I said, that virus spreads quite quickly naturally because it's got the fly vectors, and but it's not as high as the 7 kilometres we might have thought when we went into this project. So this is one of the learnings we've got from this information, but I'll tell you a little bit more about some other things we've learned. We also looked at the night counts. So these ones, they were good rabbit numbers, but a bit variable. 24 rabbits per kilometre spotlight up to 192 rabbits per kilometre spotlight. That's a lot of rabbits. So what we found after the release, we found a reduction in spotlight counts. Some of them were not really significant. Cadrona Valley, there was only a 12%, but others, they ranged between 12 and 61%. Overall, the average reduction in rabbit numbers was 39%, which married very similar to what the ECAN found, and is actually very similar to what Otago's preliminary data has said as well. What we knew, but I think we always underappreciate, is one of the things that influences impact is how many of the animals had recently seen RHDV and had strong antibodies against them, and it's very high in these areas, less so in some, but we cannot underestimate the impact that immunity plays in this virus, in the movement of this virus. Actually, there was one more thing I was going to say is, and the other problem we had with this was we actually had the uptake of the carrot bait varied considerably, and in the Ida Valley we only got 23% uptake of the carrot bait, and the others it was all over 80%. But the 23% uptake in the Ida Valley, because the Ida Valley is, you may or may not know, is usually a desert at this time of year. But look at that green grass. That was 2 to 3 to 4 centimetres high and absolutely lush. And that's just so unusual for that area of New Zealand. And the same with all of these places, had a better rainfall than they expected, and which made it for quite an unusual year. But we still did get that 30% reduction. If we look at it at the national level, so those are where we've got samples from. The yellows, the Czech, the reds are the K5, the blues are samples I haven't yet analysed as of October, and I've got samples from all over New Zealand, but I'm just showing the south so you can have a look. So we are seeing both viruses circulating, and we hope to see more. So the more samples we get in from councils, we couldn't do the study without the samples that are sent in to us. And I just summarise the impacts on those wild rabbit samples that I've just shown on that map. Again, about 2/3 were K5, a quarter to a third was Czech, and 6% was a new virus called RHDV2. And I'm going to talk that about briefly in the next slide, but basically the impacts we're seeing about, as I said, overall we're seeing 2/3 RHDV K5, 1/3 Czech, and we're seeing about 35 to 40% decrease. Anecdotally, the greatest impacts were reported in areas where there had been no recent RHDV outbreaks. And that makes sense because there'd be fewer immune rabbits. So those anecdotal impacts are often in peri-urban areas where there's not a huge rabbit population to have the virus cycling through it all the time. And the other thing I talked about, the virus movements, that was from the carcasses at the study site. But we've also got some estimates from where our carcasses have been provided by regional councils, and we have got 11.5 kilometres a month is the fastest rate of movement that we've detected. So that is within our ballpark, but yeah, and this sort of information is going to feed into some models that we're doing that'll help us answer a few more of the questions. I'll just talk briefly about RHDV2. We don't know how it got into New Zealand, we don't know how it got here. It is not the same strain as the Australians have got RHDV2 that arrived there somehow, we don't know. It's never been used as a biocontrol agent. But this is not the same as the Australians strain, and it had not been found in New Zealand until 2018. It's now the dominant virus around the world, basically. It tends to displace the number one strain, and the difference between these is RHDV1, there's about 6% difference in nucleotide arrangement, which they're all very, very similar, but when you get to RHDV2, there's been a big change. It's now only 15% similar to the RHDV1. So there has been, this is a different virus. It's not a species specific because it does kill other types of hares like jackrabbits and European hares. It also kills very young rabbits. But the big problem we have with it, it's got a very variable death rate. So until recently it was 14 to 25%, but there has been a strain that kills 75%. It doesn't still compare, because this is a naive rabbit, it doesn't compare with the 95% death rate that we get with RHDV1. It's a less stable virus, it kind of recombines. I can talk to that if you want to talk about it later. And that's one of the reasons, and the variable death rates is why it wasn't considered a good biological control agent. This finding has had a big impact, especially on rabbit owners of domestic rabbits, because the vaccine we'd been using for RHDV1 was not very good for RHDV2, and MPI had to urgently import a new one. Okay. So we've got wild rabbits in Marlborough, Flower Plenty, and very recently in the Wairarapa have got RHDV2, and domestic rabbits in Nelson and Blenheim have had RHDV2. We don't know what its impact's going to be, but some of our modelling may help us to answer those questions if we can get enough information. On the horizon, we're trying to work out what the persistence and ongoing impact of RHDV1 K5 will be. People are asking me, are we going to release any more? I think we are, in some of the, especially the peri-urban areas and and places where you can't use any other control. But I think we're going to watch and see how much it persists and how widely spread it becomes in the next year. We're going to be looking, we'd like to look at the impact of RHDV2 because we don't know what its impacts are going to be, and we don't have enough information yet, and I don't have enough resources to do RHDV2 as well as finish this project. But it really highlights the complexity of our rabbit control management and the importance of having a nationally coordinated and ongoing partnership approaches for these rabbit issues, because rabbits are a problem everywhere, but the degree of the problem varies from one locality to the other. And I just wanted to acknowledge, of course, the Rabbit Coordination Group and all the landowners, the Australian research team and our funders. Without this work would not be possible.